Dedicator of cytokinesis protein 10. Dedicator of cytokinesis protein 10 doctin, also known as zyzimin 3, is a large 240K DA protein involved in intracellular signaling networks that in humans is encoded by the DOCK10 gene. It is a member of the DOCD subfamily of the DOCK family of guanine nucleotide exchange factors, which function as activators of small g proteins. Discovery: Doctin was identified via bioinformatic approaches as one of a family of evolutionarily conserved proteins, the DOCK family. Doctin is expressed in peripheral blood, leukocytes, as well as in the brain, spleen, lung, and thymus. Structure and function: Doctin shares the same domain arrangement as other members of the DOCD/zyzimin subfamily, as well as a high level of sequence similarity. It contains a DHR2 domain that is involved in G protein binding and a DH-RON domain, which in some DOCK family proteins interacts with membrane phospholipids. Like other DOCD subfamily proteins, doctin contains an N-terminal pH domain, which in DOCK9/zyzimin mediates recruitment to the plasma membrane. The DHR2 domain of doctin appears to bind to the small g proteins CDC42, TC10, and TCL, although these interactions are of low affinity. The physiological role of doctin is poorly characterized. However, a study in lymphocytes has shown that doctin expression is upregulated in B lymphocytes and chronic lymphocytic leukemia CLL cells in response to the cytokine IL-4. This suggests that doctin may have a role in B cell activation and proliferation. Another study identified doctin as a protein that was overexpressed in some aggressive papillary thyroid carcinomas.